Just want to get it out of the way. Thank you.
Good morning, everybody. Welcome to MSU and Sparrow Pediatric Grand Rounds and to the very first session of 2018. I hope you all had a very nice break. I'll also like to extend a warm welcome to our colleagues in Flint and Grand Rapids. Before we get started, I have a couple of announcements. Today at Subspecialty Rounds, Dr. Mora will do a cardiology presentation. Please join us next week when Dr. Goebel presents Pediatric Syncope, Beyond It's Not Your Heart. I'd also like to draw your attention to evaluations that would have been in your inbox about Grand Round sessions. We value your feedback because we're going to use it to improve future sessions. It is my distinct pleasure and great privilege to introduce this morning's speaker, Dr. English. And even though Dr. English is very well known to all of us, there are a few things about him that some of us don't know. Dr. English is professor and chair of the Department of Pediatrics and Human Development here at Michigan State University, and the academic chair of the Department of Pediatrics at Helen DeVos Children's Hospital of Spectrum Health at Grand Rapids in Michigan. Dr. English completed his undergraduate degree in biology at Cole College, Cedar Rapids in Iowa, where he graduated summa cum laude. He obtained his medical degree at Baylor College of Medicine in Houston, Texas, and completed his internship and residency in pediatrics at Texas Children's Hospital and Ben Taub General Hospital in Houston, Texas. He was a research fellow in the Division of Infectious Disease, Department of Pediatrics, University of Washington Children's Hospital and Medical Center in Seattle, Washington. Following his completion, the completion of his fellowship, he joined the faculty at the College of Medicine at the University of Tennessee Health Sciences Center in Memphis as an assistant professor of pediatrics and the Department of Biochemistry at St. Jude Children's Research Hospital in Memphis as a visiting scientist. He subsequently rose to the rank of professor and chief Division of Infectious Diseases at Lugano Children's Medical Center. He joined the Department of Pediatrics and Human Development here at Michigan State University in 2013. And since then, he's been really involved in resident education and several activities within our community, especially trying to bring all the community centers together and certainly fostering a lot of advancement in research. And precision pediatrics has been his baby. Dr. English has authored several peer-reviewed publications and written several book chapters and reviews. He's a reviewer and the editor for several peer-reviewed journals. He has also won several honors and awards, including resident teaching awards, which is not surprising. He's won while he was in Tennessee, he had an award from the Tennessee Hospital Association, Memphis Business Journal, Healthcare Heroes Award, Pediatrician of the Year, and several other honors and awards. He's a funded investigator with several past grant awards, including NIH grants, even though now he's got a major administrative role. He has no conflicts of interest to report in the preparation of this talk. Please welcome Dr. English as he presents Is Precision Pediatrics Really Possible?
Thanks so much. Thank you. Thank you for that kind introduction. And welcome everybody and Happy New Year. This is one of my favorite topics, and I am going to talk about it today. A little bit different spin than you may have heard me talk about it before. So, can we really do precision pediatrics? That's the topic of the talk today. I have no disclosures or conflicts of interest, but I do have a couple of slides I can't resist showing since I was a long-suffering Houston Astros fan since I grew up in Northeast Texas many years ago when they were called the Colt 45s. I had the opportunity to go to Game 5 of the World Series in Houston. That's Shelley Kaplan there, one of the leading lights in pediatric infectious diseases, one of my mentors at Baylor when I was a resident there. And we managed to attend Game 5, which the Astros won, and went on to win the World Series. So I couldn't resist showing those slides today. If you didn't see the cover of Sports Illustrated, 4 years ago, they predicted that the Astros would win the World Series in 2017. And you may remember Yogi Berra once said, the problem with most predictions is they're difficult to make, especially about the future. And most predictions Sports Illustrated has made are wrong, but this one was right. So let's talk about precision medicine and precision pediatrics. The critics of precision medicine, I don't see Dr. Fantha here, he's one of the most vocal and well-published critics of the idea of precision medicine, argue with some validity that precision medicine to date has largely been boutique medicine, or as they say in Michigan for reasons I don't understand, boutique medicine. That it's just a way to provide a diagnosis for that rare patient with a rare condition. And if that's all precision medicine amounts to, it will never revolutionize healthcare. It's got to be something that will allow us to address common problems that are major problems that affect a lot of children and their families, and of course adults and their families. Things like autism, asthma, obesity. So for precision pediatrics to succeed, it's going to have to improve the health and well-being of populations of children, not just of the child with the rare condition. It's hard to believe this now, but it was almost 3 years ago when President Obama had a press conference and at the State of the Union address then talked about his new precision medicine initiative. He said, tonight I'm launching a new precision medicine initiative to bring us closer to curing diseases like cancer and diabetes and give all of us access to the personalized information we need to keep ourselves and our families healthier. Pretty ambitious. And it's always ambitious when you use the C word, cure, curing diseases like cancer and diabetes. Of course, it's most likely that the impact of precision medicine is going to be to ameliorate these conditions in most cases before they're cured, but nonetheless, it's a good goal. So why this term precision medicine or precision pediatrics? The literature often includes the term personalized medicine. You'll see individualized medicine or precision medicine. Well, the reason that precision medicine won out after a lot of discussions at the national level is too many people thought personalized medicine sounded like the Jetsons. You go to see your doctor sequences your genome in the back room, plugs things into the computer, creates some new drugs for you, sends you home with your prescription, 5 minutes later. That degree of personalized medicine ain't happening for a few years yet. But what could happen in precision medicine would be tailoring medical treatment to individualized characteristics of individual patients. That's been done and is being done. And in fact, some of the skeptics or cynics about precision medicine say, well, what do you think we've been doing all these years? Have we been, and I'll show you a slide or two to say, well, when you have trouble seeing and you go to see the ophthalmologist or the optometrist, they don't just take a random, pair of glasses off the shelf and hand it to you and say, I hope this helps. They refract your vision and give you the eyeglasses that will correct the problem. Blood transfusions, we've known for many years what the major and minor blood group antigens are. And we know that if you're in an accident and you need a pint of blood, we need to type and cross you to give you the correct type of blood. However, this idea that we could use really big data and connect all sorts of data from health records, from social media, from genomics, etc., and use all of that to affect the health care and the health and well-being of individual patients, that is a relatively new idea. So here's the bias. Here's the advertisement. The next few lines are what President Obama's task force and the NRC said we needed for precision medicine. They said, well, we need really some rigorous research to provide good evidence to turn this concept into reality, not just a few case reports or a random case series. And we're going to need a lot of bright people who want to change the world to do this, to join the research team. We have to figure out a way to standardize these data so that they can be shared by other people, because they're not much value if the data that we get from a cohort here isn't representative of another cohort. You got to figure out a way to connect these dots and share the data, and that's what the Health IT Precision Medicine Task Force said 2.5 years ago. And they also went on to say, the same task force said, you know, let's do this. It's the time to do it now. We have the science is coming. We have the tools to analyze these data. We've got new techniques that are going to get better and better. We're going to be able to connect the dots. Let's do this thing. So the promise of precision medicine in pediatrics is great. And, you know, when I'm talking to a pediatric audience, it doesn't matter. But I've talked to many mixed audiences, including lay audiences, about this idea of precision medicine. And I don't need to tell anybody in a room of pediatricians that if precision medicine is going to work, it's going to have to start with pregnancy and infancy. You know, precision medicine, I'm all for precision diagnostics and treatment of adults, for example, with cancer. That's great. But to really impact health, we need to prevent diseases, and that's going to have to start with a precision medicine in pregnancy, infancy, etc. And to quote one of America's least articulate presidents, I never thought I would miss him, but I do. One of America's presidents once said, if we do not succeed, we run the risk of failure. You can probably figure out who that was. So what is precision pediatrics then? Well, the idea is simple. It's identifying specific children who are at risk for specific problems and who would benefit from specific therapies, but not benefit from, and in fact, not be harmed by other therapies. One aspect of precision medicine that's probably not been emphasized enough, in fact, the early successes in precision medicine had to do with preventing the administration of a therapy that wasn't going to work and was just going to be harmful. I'm thinking now of sort of pioneering work done at St. Jude years ago when they figured out that something like 1 in 150 kids do not metabolize 6-mercaptopurine. If you give them the standard dose of 6-mercaptopurine, they'll get severe toxicity and might die. And so now every kid in America who's going to get that drug for cancer is screened first to find out if they have a mutation that prevents them from metabolizing this drug. So the idea about precision works both ways. It's let's not give you something that you're not going to benefit from but that might harm you, and let's try to target therapies for you that really would work. I'm not going to talk about it anymore today, but one of the biggest potential opportunities here is in the treatment of behavioral health and psychiatric conditions such as depression and ADHD. All of those of you who've cared for children or adults with these conditions realize how frustrating it can be to know that a patient is likely to benefit from one of several drugs, but not knowing which one is going to be most effective. Often having to start with a low dose of a medication, increase the dose gradually over time until either you see some benefit or side effects, give up on that, start another drug, do the same thing again. Obviously, the goal would be to determine not only to have precision in your diagnosis of these conditions, but to be able to identify up front which of a particular group of therapies is likely to benefit that patient. That's the goal of all of medicine. Obviously, this is the right time to do it. The revolution in biological science in the last few years has made it possible to dream about doing things that were not dreamed about much when I was in medical school not that long ago. And of course, it does start with nucleic acid sequencing and technology. The critics of precision medicine say, well, genomics will never be the answer to all of our medical problems. And of course, they're right. But it is a big part of it. It is part of the big data that you need to sort this out. I'm required by law to show this slide in any talk about the impact of genomics on medicine. And it's an important slide. Do people here know what Moore's law is? Moore predicted, I guess 30 years ago, that computers would continue to get faster and faster and faster. The amount of computing power you could put on a chip would double every couple of years, every year and a half or 2 years. And Moore's law has pretty much been true to date. So computer power gets faster and faster and faster. Eventually, Moore's law, though, will peter out because once you get down to the smallest molecular computer processors, there'll be a limit on how fast computers can be. They'll be very, very fast. They'll be much faster than they are today. And that Moore's law, as I say, has been pretty much valid. If you look at the early days of sequencing the genome and how much it costs to sequence a genome, you can see that it sort of followed the trajectory of Moore's law. The first genome that was sequenced cost somewhere between $75 and $100 million, probably, if you count up all the work that went into that. And over the years, that got cheaper and cheaper and cheaper. And while there's no, I notice now that this version of the slide doesn't have the years down there at the bottom. A few years ago, sequencing technology improved so rapidly that it improved much faster than computing power did. And this turns out to be important because we can now sequence a genome for probably $1,000 or less, but to sequence and interpret the genome costs much more. And in fact, there's a disconnect between the amount of data we can generate from sequencing and other forms of connecting big data, and how long it takes us to analyze and understand that data. And that has caused its onset of problems. This is Shenzhen, China, where I was at the World Society of Pediatrics a few weeks ago, and I put this slide in only to remind me that about 10 years ago, or maybe 7 or 8 years ago, Beijing Genomics Inc. in China, which still exists and has I think a couple of hundred sequencers. Chris Contag, I saw in the audience, I can ask Chris, he probably knows how many they have. They've got all these sequencers working around the clock. And about 7 or 8 years ago, they were generating so much genomic data that they didn't have enough computers to store them on. And there was a story in the New York Times that if you sent information to them to sequence at one point, they were sending you back your sequencing data on a bunch of CDs. Well, it's not very helpful to have a bunch of CDs that say A, C, T, G, G, T, A. See, that just wasn't very helpful. It just helped illustrate how the ability to even store the data was limited at the time, much less analyze and understand all of that data. And believe me, the genomic data is only one part of this big data. So we're going to talk a little bit about omics. So omics are broad disciplines that are based really on the idea that you only understand data when you understand how it interacts. It's not static. Genomics, of course, is the omics that really led to the idea of precision medicine. And it's much more ambitious than traditional genetics that look at the effects of a particular individual mutation. So genomics seeks to understand how genes interact with each other, with the rest of our DNA, the non-coding reasons. It's linked to epigenetics, which we'll talk about briefly in just a minute. So there are a lot of omics. There is genomics. The transcriptome really is the totality of all the messenger RNA in the cell. So those are the genes that have been transcribed into RNA. The epigenome is everything in the genome that is telling the genome when to be turned on or not. And the epigenome and epigenetics is where nature meets nurture, right? I mean, it's the environmental effects, whether it's stress or a pesticide or a hormone that affects whether a gene is turned on, when it's turned on, how long it's turned on, which transcripts are created, etc. So the transcriptome is there because the epigenome has interacted with the genome. So it's how the environment modifies gene expression, and again, where nature meets nurture. Then there are proteomes, metabolomes, interactomes, kinomes, and don't forget about the microbiome, which we'll come back to a little bit later. We have to remember that microbes outnumber our human cells by about 10 to 1 in our body. We can't forget about that. So if that sounds complicated to think about wrapping your brain around all these different omes that are going on, how about trying to monitor that in real time? Measure all of that, do something, measure it again, see what happens with all of these complicated interactions in the course of a disease, for example. Well, that's what George Mias has been doing. He's been working on what he calls dynamic omics. And I borrowed a few slides from George that I'll show you about some of his work that he did, pioneering work he did at Stanford before he was recruited to come here to MSU. So dynamic omics is this one way to get toward real personalized medicine. And what it is treating all those different sets of related data that I mentioned, the transcriptome, all the RNA, the metabolome, all the metabolic products and processes in toto, the proteome, the microbiome, the genome, the methylome, which is part of the epigenome, part of the gene expression is controlled by whether certain residues are methylated or not, an autoantibody home, etc. George says, let's treat all these things like pieces of a puzzle, and then let's measure them all and put them together. And so you collect samples, you measure all of these things, you generate terabytes of data on individual patients, and at the end, you try to integrate all that to see how it differs between people and how it changes over time. So George did a landmark study that he was a co-author on, published in 2012, doing this over a year with the head of genetics there at Stanford, who really was the patient, the quote patient for this study, I think in part because they realized it would take forever to get IRB approval for anybody else to be the patient. So he was actually monitored over a long period of time, and one of the things they discovered was he actually developed type 2 diabetes during the study, and they were able to analyze some of the data they'd gathered before that to help figure out how that could have been predicted. Anyway, the bottom line is, and I won't share any more of this data today, is this idea of quantitatively being able to assay things like this over time is something that George continues to study. He's interested in how this might, what might happen during an infection. During the more than a year they followed Dr. Snyder, he did develop a couple of respiratory viral infections, for example. So they were able to have some samples to study before and after. You can imagine what kind of data, the amount of data that might be generated from such studies, and the need for more and more powerful techniques to analyze that data to figure out what it all means. Back to Shenzhen, China, in the Bougainvilleas. So why is MSU investing in precision medicine research? Well, because we need a precision medicine approach if we're going to improve population health. You know, MSU obviously has this community campus footprint. We have 7 major campuses across the state. We have great needs in the Upper Peninsula and Traverse City, Flint, Southfield, Midland, Lansing, and Grand Rapids. So a precision medicine approach really, I think, is in the long run the way you improve population health. It's also the way to do important research that's likely to get funded in the next few years. It fits with grant initiatives and with NIH funding priorities, and certainly for those of us in pediatrics, NICHD priorities are very relevant. And the NICHD has said that they want to support research that will catalog and identify interrelated environmental and genetic factors that are key to health. That's the epigenome, that's the genome connected to the environment. In particular areas they're interested are in developmental origins of health and disease, behavior and cognition, including interventions for autism and pharmaceutics for brain recovery in people who've had a brain injury. It fits with the Brain Initiative, and obviously it fits with the National Human Genome Research Fund. It fits with this idea that we've had in pediatrics since before I came, and which I have embraced, which is we need to have partnerships across multiple communities. They mentioned that in my introduction. So it fits with things we can and should be doing together. For example, in Grand Rapids, it fits with this expansion of pediatric neuroscience, clinical programs in neurology, epilepsy. It fits with research at the Van Andel in epigenetics and neuroepigenetics. It fits with autism-related research in pediatrics here, and it fits with the public health expansion in Flint because neurocognitive development is a major area of concern in the high-risk population of kids in Flint. It fits with the new research buildings and institutes that are present at MSU today, the IQHSC building that Dr. Kontag runs and right next to Life Sciences. It fits with the new Grand Rapids Research Center that's just opened in November in Grand Rapids. And I think all of you have noticed the cranes next door to the IQ building in Life Science. This building effectively is being cloned, and a version of this building is being built right now here in East Lansing next to the IQ building. And that's going to be an integrated interdisciplinary science building that will feature quite a bit of neuroscience. So for a variety of reasons, it's the right time to do this at MSU. So how do you do it? How do you build precision pediatrics or precision medicine? I think you have to do 3 things. You've got to have outstanding scientists who study omics, understand, and know how to analyze big data. You really need physician scientists as part of this team. I really think this is more important than ever, and it's harder and harder to get physician scientists, young physicians in training to commit to careers in research. But we need physician scientists as part of the team. In particular, we need physician scientists who really have a goal of improving a particular condition. I think over and over and over, look in the history of medical research, outstanding advancements have been made. I think of my friend John DiVincenzo in Memphis, who has really led the way in developing new approaches to treat RSV infection in children. This is something John's worked his entire career on. You need people with that kind of fire in their belly that want to change the outcome for kids with autism or asthma or epilepsy. And eventually you need health services researchers. If we do all of this and we find new great things that we could apply to populations and we never apply them to populations of patients, we failed. So I'm going to switch gears a little bit and talk about something I haven't talked about before in this forum. A little bit about how precision medicine should revolutionize the field I trained in, pediatric infectious diseases. I think it'll be the key to preventing infectious diseases in the future, and it's going to be a way to improve outcomes for children with infections, including congenital infections. We need precision medicine approaches to explain the variation in infectious disease susceptibility and severity. When I was a medical student, I asked my attendings those annoying questions like, why did this child get pneumococcal meningitis? And my really smart teachers, like Dr. Kaplan, said, well, you know, the incidence of pneumococcal meningitis in kids between 6 and 12 months is blah. You know, here's the data. We know what the incidence is this, the incidence is higher. In the first, it's higher between 6 and 12 months, and then 12 to 18 months. It goes down after 2 years. We knew what these curves looked like. We knew who was at greatest risk. We also knew about something that we called immunodeficiency. We knew that there were children that had severe combined immune deficiency. At the time, we didn't know that that was a mutation in the common cytokine gene receptor, but we know that now. We knew that kids, some little boys didn't make antibody, and they got what we called Bruton's agammaglobulinemia, named after Colonel Ogden Bruton, who described this. We now know that's a mutation in a gene that has to do with B cell development called BTK. We knew there were a few of these immune deficiencies, but really, we didn't know why other kids got it. So many kids got exposed to meningococcus, for example. We've known for many years that if we culture everyone's nose in this room, we would find that a few people are harboring meningococcus in their nose. Thank goodness most of you would never ever get sick from that meningococcus. But we don't know why that is. We don't know why of all the people exposed to germ X, only a few get disease. And when I asked about that as a medical student, I got those numbers like, well, the incidence is blah, or I got what basic scientists call hand waving. Somebody tried to change the subject to tell me about something else because they had no idea why this little boy got meningitis. And that's what parents want to know, right? Parents don't want to know what the incidence of pneumococcal meningitis is. They want to know, why did my child get pneumococcal meningitis? Or why did my child get this even though we gave them the vaccine? Why do some kids fail the vaccine for common childhood diseases? Well, the bottom line is almost certainly it's due to small genetic differences between people that affect the way that we respond to pathogens and the way we respond to infectious diseases. And I'll tell you a little bit about how that was discovered over the past 10 or 15 years as an example. What I call lacunar immunodeficiencies, by that I mean these are people whose immune system is pretty normal. And they're going to be perfectly fine unless they're exposed to germ X. And if they're exposed to germ X, they might get severely ill and they might die. We've known about something like this for many years. We know that some little boys with their primary infection with EBV infection, Epstein-Barr infection, the virus that causes mono, some little boys get a terrible lymphoproliferative disorder and die from that. We know what the genes are that cause that. Now, we've known forever it was X-linked. So we've known about examples about this for years. Those little boys are totally healthy, and if they're never infected with EBV, they'll have a pretty normal life. But if they're exposed to EBV, they're in trouble. Well, there are a whole bunch of examples we know now where genes modify your susceptibility to disease and the severity of disease. They're not the same thing. Something that increases your risk of developing a disease might or might not increase the risk that you will have more severe disease when you get it. So they're not even the same genetic factors. So let me tell you this little story about innate immunity and how the discovery of innate immunity and how this led to some of our improved understanding of genetic variability and how that affects infectious diseases. So I trained in the 80s in Chris Wilson's lab in Seattle studying macrophages, one of the key white blood cells that are involved with innate immunity. And at the time, it really wasn't very sexy to study innate immunity because everybody wanted to study T cells and B cells because a lot was known about them and more information was being learned. It turned out that innate immunity is required to instruct T and B cells when to do things. But we didn't know that at the time. Well, in 1989, Charles Janeway at Yale, the son of a very famous Dr. Janeway, who you know about, who had been at Harvard, who described those Janeway lesions and infectious endocarditis and a variety of other things. Dr. Janeway proposed, reportedly at a session in the bar after a conference at Cold Spring Harbor when people were just talking over a beer, he said, you know, there's got to be something about microbes that alerts the body that there's danger. There's got to be some kind of danger signal. Everybody knew a lot about what happened in the immune response to pathogens, but people really didn't understand how does the body know that this germ is a problem in the first place? Because we're bathed in germs all the time. A lot of bacteria live in our body and aren't causing harm and maybe doing some good. Why is it that a certain germ is recognized as being dangerous? So he said there's got to be some kind of, quote, pattern recognition receptor. They've got to recognize something on the surface or something secreted by a pathogenic bacteria, for example, that's different than what's on a non-pathogenic bacteria. There've got to be these pattern recognition receptors. Another name for them that became popular in the literature was PAMP, a pathogen-associated molecular pattern. Again, the idea is something that's different in a pathogen than in a commensal organism that doesn't cause disease. It's got to be something important if the body's going to be able to recognize it, because if it's something that the germ doesn't need, we know bacteria multiply rapidly and they're quite capable of mutating and losing something that's not important. It can't be present in the host, or we wouldn't likely have an immune response to it. And one obvious pattern to check out would be endotoxin or lipopolysaccharide, because it had been known for many years that part of the problem with severe gram-negative infections, such as gram-negative sepsis or meningococcemia, is that bacterial endotoxin, which is a lipopolysaccharide, triggered this massive immune response that caused harm to the body at the same time the body was trying to kill the pathogen. Bacterial DNA was another potential PAMP to study. And so the idea was something in the bacteria has to sound this danger signal. So the way that it was discovered what the main pattern recognition receptors were for bacteria in humans was really by chance, and really helps illustrate the value of why we must continue to invest in basic science research. We can't only invest in research that's targeted toward a particular translation that we already think we know how it's going to work. Jules Hoffman and colleagues in Strasbourg, France, were studying fruit flies. And they knew that there was a protein called toll in fruit flies that determined whether or not the fruit flies developed their abdomen the way they should, the embryonic development. Because if they didn't have this toll gene, they never ventralized the abdominal pattern. It's remained all on the dorsal side. And if they had a gain of function mutation, they ventralized too much and they lost a lot of the dorsal pattern. So segmentation of development of the abdomen and thorax in fruit flies, something that nobody thought had anything to do with immunity, right? So they were studying this, busily studying this, studying all these fruit fly mutations. By the way, toll, I'm told, is sort of a mild expletive in German. Somebody was actually looking under the microscope at the first fruit flies that had this weird pattern of their abdomen and said toll, and that became the name for the mutation, way before anybody studied a role in immunity. So it turned out, though, that toll did have something to do with innate immunity in fruit flies. And fruit flies don't have adaptive immunity. They don't have T and B cells, but they do have primitive innate immunity. And it was known already that host defense in fruit flies depended in part on production of these peptides that had antimicrobial properties that were made by the fat body in the fruit fly, which functions a lot like our liver does. That was known. One of those proteins had been identified and was called drosomycin. People knew that that was an important antifungal peptide to help prevent fruit flies from being infected with fungi. And so what Jules Hoffman and his group did in Strasbourg was sort of a low throughput screening. They just checked a whole bunch of cells from mutant types of fruit flies, stimulated them, and measured whether they made drosomycin or not, just like on a little dot fly. And one of the ones that was negative was toll. So the flies that had a mutation in toll didn't make any of this drosomycin. And they thought, well, that might be important. So maybe the toll protein has something to do with immunity in fruit flies. So they did this experiment, which has one of my all-time favorite experiments and one of the best covers of Cell ever. In 1996, and actually I heard them present this data at a Keystone conference before it was published, they did the following experiment. They took fruit flies that either did or did not have a mutation in the toll gene, and they infected them with aspergillus. Now, the way they did it is pretty cool. Fruit flies aren't stupid, so if you put them on a petri dish and you had aspergillus, guess what? They fly away. So they had to have a petri dish that had sort of a stickum on it, the way that you would, catch a fly out on your fly trap out in your yard, or a mosquito. And so they had to stick their feet to the petri dish so they couldn't fly away. Then they added aspergillus. And the normal fruit flies didn't matter. They were fine. They couldn't fly away, but they didn't get infected. The fruit flies that had a toll mutation, this is what happened to them. That's not a sweater. That's aspergillus that is killing this fruit fly. So that's a dead fruit fly, and what looks like a sweater is just a thick mat of aspergillus. These fruit flies died of overwhelming aspergillus infection. And when they reported that, immediately they thought, and other people thought, well, gee, I wonder if there's something like that that controls immunity to fungal infections in people, or bacterial infections. So now it goes back to Dr. Janeway, who, Dr. Janeway, by the way, sadly died of a brain tumor about a year or two before he would have won the Nobel Prize, because he and a very talented postdoc of his, who's now one of the leading immunologists in the world, Ruslan Metsitov, found that there were toll-like receptors in mice and in people. Ironically, they're quite different in the way they're structured, but the bottom line is they were cousins phylogenetically. So there were toll-like receptors in people and in mice. They reported that in 1997. And then a few months later, Bruce Beutler reported that one of those toll-like receptors, the one called toll-like receptor 4, was that sensor for endotoxin, or LPS. People have been looking for the receptor for LPS for 40 years, and nobody had found it. But it was this discovery, chance discovery in a fruit fly, that led them to look for things that were cousins of toll, and they found these toll-like receptors. They actually work differently in humans and in mice than they do in the fruit fly. In the fruit fly, toll is a secreted protein that binds to a secreted protein called spatzel. And in man and in mice, the toll-like receptors are either on the cell surface, like you see toll-like receptor 4 there in green binding to LPS, or they're inside in an endosome, like the receptors that bind to bacterial or viral nucleic acids, toll-like receptor 9 or 7 or 8 or 3. They're all in the endosome, whereas the other toll-like receptors are on the surface. So it was quickly understood how many toll-like receptors there were. People figured out what the signaling pathways were downstream of the toll-like receptors. They're the most complicated downstream of toll-like receptor 4, which has 4 adapter proteins. Other toll-like receptors use some, but not all of those adapter proteins. So it's complicated, but the bottom line is, in all these cases, these toll-like receptors recognize some bacterial products. That triggers an inflammatory response, production of cytokines, other mediators like nitric oxide, and that turns out to be really the most important of all of the danger signals in the human body for recognizing pathogens and mounting this response. So you won't be surprised to learn that if this system is that important, then mutations in the signaling pathways might be a problem. And I'll show you that in just a minute, or maybe I'll show it to you here. The double-stranded RNAs, like you would see in replication of herpes viruses, for example, signal through toll-like receptor 3, other viral RNAs through toll-like receptor 7 and 8, and then bacterial DNA through toll-like receptor 9. And that'll suffice for what we're going to talk about today, except let me call your attention to the blue, dark blue on the right side, flagella. Actually binds to toll-like receptor 5. Now, the flagella from bacteria is largely a monomer of thousands of copies of this particular protein, flagellin. And flagellin is recognized by a toll-like receptor 5. I'll come back to that in just a minute. So these toll-like receptors were discovered. Unfortunately, Dr. Janeway had died, but Dr. Beutler, who had discovered toll-like receptor 4, was the ligand for LPS, and Dr. Hoffman, who did the original studies in the fruit fly implicating toll immunity, shared the Nobel Prize in 2011 with Ralph Steinman, who got it for a different type of research on dendritic cells. Dr. Steinman is the sad answer to the trivia question of which Nobel Prize was awarded to someone who was deceased. Dr. Steinman was alive when the award was decided upon, but he died before the ceremony. So Nobel Prizes are not given to anyone who is deceased. So Dr. Steinman won it for dendritic cells, Dr. Beutler and Hoffman for the toll-like receptors. So as I said, you won't be too surprised to learn that if these are important sensors, the mutations in these proteins might be associated with a change in susceptibility to human diseases. There's a ton of data about this now showing that this is true. Everything from the fact that small polymorphisms in some of these proteins are associated with an increased or decreased risk of certain diseases. And by the way, the risk is often unpredictable, and you don't know exactly what the risk would be until you study it, to situations where you have larger mutations, even absence of some of these proteins that are associated with a particular phenotype or disease. And to me, still the most interesting one is this story of toll-like receptor 5 in Legionnaire's disease. So Legionella pneumophila is an unusual flagellated gram-negative bacterium, which we know causes outbreaks of pneumonia due to contaminated water supplies. The original outbreak in the hotel in Philadelphia during the Legionnaire's convention is why it's called Legionnaire's disease. Interestingly enough, disease caused by this agent had been described previously as a flu-like syndrome in Pontiac, Michigan, Pontiac fever. So this is a disease that's not just from contaminated water, but that's where outbreaks occur. It's almost always contaminated water supplies. I mentioned a case in Philadelphia, fascinating case we had in Tennessee when I was in Memphis. A colleague of mine who trained with me at Baylor discovered in Knoxville there was a cluster of people with Legionnaire's disease, which she traced to a local restaurant which had a fountain in the foyer. And it turned out that people that went on busy nights sat in the lobby longer and were more likely to be exposed to the Legionella that was in the water that was being aerated around the room, and were more likely to get Legionnaire's disease. I love epidemiologic studies like that, such as those studies that show you're more likely to get certain diseases that are waterborne if you were in a triathlon and you're really a bad swimmer. If you're a bad swimmer, you stay in the water longer and you swallow more water, you're more likely to get disease. Leptospirosis, for example. Anyway, so toll-like receptor 5, what's the story then of toll-like receptor 5 and susceptible to Legionnaire's disease? Well, a really smart basic scientist in Washington named Alan Aderum thought, well, gee, if toll-like receptor 5 is important in responding to these flagella, then it ought to increase the risk of some of these diseases caused by flagellated bacteria like Legionella. And Legionella, he reasoned, that's something I can study because there tend to be outbreaks of Legionella, unlike a lot of these other diseases, outbreaks around water. So there was a big outbreak of Legionnaire's disease in 1999 in the Netherlands in association with a flower show. If you've ever been to a flower show, and I've been to the world's largest one in the Netherlands, it's like walking through a rainforest for hours because they've got to mist all those orchids and everything or they will die. So you're walking through misty water the whole time you're in the flower show. Well, there was an outbreak in 1999. They quickly showed, yeah, the water is contaminated, the sprinklers are contaminated, the whirlpool spas are contaminated. And so what Alan Aderum did was he went to the Netherlands. He studied people who had been diagnosed with Legionnaire's disease. He found controls who had similar amount of exposure at the water show, and then he sequenced them to see if they had abnormal versions of toll-like receptor 5. And what he found was they were more likely, the people who got disease were much more likely to have a truncated form of TLR5, but they had no expression on the surface at all in many cases. And they had Legionnaire's disease, and other people at the flower show had been infected, because you could prove that serologically, but had not been sick. Other people weren't infected at all. And I don't know if he's ever sorted out what the difference was between the people that didn't get infected versus those who got infected but didn't get sick. But certainly the ones who were ill were more likely to have this truncated form. They were otherwise healthy people. They did not have what we would consider to be a traditional immune deficiency. And it's obvious that if you knew who these people were and you had a vaccine for Legionella, they're the people you would give it to, right? I mean, you're not interested in immunizing the population for Legionella, most likely. It's not very common. It wouldn't make sense. But it would make sense to immunize these people if you knew who they were. So there's toll-like receptor 5 over there again. Well, how about other lacunar immunodeficiencies and toll-like receptor signaling defects? Well, there's an investigator, and he was in Paris, he's still in Paris part-time, called Jean-Laurent Casanova, great name for an investigator, who spends half of his year at the Rockefeller University in New York and half the year in Paris now. He described patients, 15 years ago, with a deficiency in one of those signaling proteins that's downstream of the toll-like receptors called IRAK4. And these patients got recurrent invasive infections caused by pneumococcus and Staph aureus. And that's an unusual phenotype. For the infectious disease docs and immunologists in the room, you know that patients that get recurrent or severe pneumococcal infections tend to get infections caused by other encapsulated bacteria like Haemophilus or Meningococcus and tend to have a problem either making antibody or complement. Whereas patients that get staph infections those patients with antibody deficiency don't get staph infections. People that get staph infections usually have a problem with their neutrophils or with opsonization or both. Well, it turned out IRAK4 mutation downstream of the toll-like receptors, maybe not surprisingly since so many different toll-like receptors use it, was associated with infections caused by both of these. There's IRAK4 there downstream of all the toll-like receptors and also some cytokine receptors and where its signaling goes. And so he figured that out quite a while ago. Dr. Casanova, by the way, is the most either confident or contrarian of all of the immunologists who study what I'm talking about today. He's the one who says out loud, I think all of the variability in human disease, infectious disease, is caused by these small genetic differences between people, all of it. Virtually none of it is caused by differences in the pathogen. That's probably not totally true, but it may be largely true. For example, for RSV, for many years, we've tried to figure out, are there some strains of RSV that are much more severe than others? There are 2 different main types now, but nobody really has any good evidence that you're more likely to get severe disease because you got RSV strain 1 or 2. It's mainly about the host. Even the inoculum doesn't affect it as much, probably, as the host differences. Anyway, Dr. Casanova is certainly the true believer, and he's got a lot of other ammunition. This might be some of his very best data, is for herpes encephalitis. So outside the newborn period, we know that newborn babies are particularly susceptible to herpes, and we think we know why. They don't make much gamma interferon, they don't have good communication between their T cells and their antigen-presenting cells. We think we understand why a newborn, or particularly a preterm newborn, exposed to HSV is more likely to get disseminated disease and maybe encephalitis than a 2-month-old. However, we don't really understand why most people get herpes encephalitis. This is a relatively common form of encephalitis. It can be fatal. It's certainly severe. It's one of the few that we have a good treatment for these days. But for many years, we've tried to figure out, why did this patient get herpes encephalitis? It's not something we see necessarily in immunocompromised patients. Out of the blue, we see herpes encephalitis. We know that in many cases, it's not even the primary infection with HSV-1. Or even 2. It's usually HSV-1, but it's not even the primary infection. They may have been infected years ago, have a recrudescence now, and they get encephalitis. Why is that? I don't know. No one's ever been able to figure that out. That's one of the, why did this patient get herpes encephalitis, considering that half the general population or more has been infected with HSV type 1? Why do so few people get encephalitis? Dr. Casanova wanted to know that. And so he found an autosomal mutation in 2 children with HSV encephalitis that appeared to explain it to them, and they found more mutations later. I'm sorry I'm missing the slide that tells you what his strategy was. What he did was, in France, they have a registry of all the patients with herpes encephalitis in the country, everybody. So he was able to get that entire registry, and he looked for cases where there was consanguinity in the family, because he knew if there was consanguinity, that would increase the risk that an autosomal recessive mutation might be shared and might cause such a disease. And I don't remember what the number was, but it was something like compared to 2% consanguinity in the population. There was 12% consanguinity in this cohort of kids with HSV encephalitis. That already told him he was onto something. So he found those families. He did a lot of sequencing. He looked at the toll-like receptors. And sure enough, right away, he found a mutation in one of the signaling proteins downstream of particularly toll-like receptor 3 called UNC93. It doesn't really matter the name of the protein. It's in that signaling pathway. And by that time, he and other people had developed strategies to quickly test white blood cells from a patient to see if they responded to these different ligands or not. So he knew that's what it was. So he found that. Then he found 2 more children with a mutation in the toll-like receptor 3 itself. And now since then, he's found a number of other mutations. You've seen UNC93B up there. He's found other mutations downstream of that and in toll-like receptor 3. He thinks, and he hasn't proved this yet, he thinks he'll eventually find some sort of mutation or polymorphism in every patient with herpes encephalitis that will explain why that person got this relatively uncommon condition. The skeptics say, no, it accounts for some of it, but a lot of it's just that variability that we've always seen in infectious diseases. And maybe they're right, maybe he's right, I don't know. So summary is that an improved understanding of innate immunity infectious agents should lead to more effective prevention and therapy. These sort of discoveries beg the question of what should immunization strategies look like 20 years from now? Should we be sequencing kids at birth and determining based on their sequencing, this is a child who needs a mutation against Legionella. Here's a child that needs a different immunization against pneumococcus because they're not going to have the optimal response to this vaccine, or they need the meninge B vaccine, but they need a different adjuvant because their genetics tells me they're not going to be able to respond to this vaccine. That is likely the future of immunology and infectious disease in terms of prevention. The other side of this, and I don't have the slides in here today, is on the vaccine development side. This is revolutionizing vaccine development, so-called reverse vaccinology. The way that vaccines were traditionally made was somebody took serum of somebody who had survived an infection. They took that serum to see if it would prevent mice from getting a disease caused by that pathogen. And if it did, they tried to figure out what it was, and what it was often an antibody. So they figured out, okay, here's an antibody against X. That's how Father and Gill and Wright did the classic studies of Haemophilus influenza type B that led to a HIV vaccine. So we figured out that antibody, what it was against. That was the vaccine we needed. We finally made it. Reverse vaccinology starts with the bug. We just sequence the bug. We look to see what proteins are predicted to be expressed on the surface. And then we choose some good candidates and we make vaccines out of them and we test them. That's how the meninge B vaccine was developed. I don't know if you know this, but the reason that we had a meningococcal vaccine for groups A, C, Y, and W135 and not for B was that the polysaccharide on the surface of B is the same polysaccharide that's on the surface of some of our cells in our brain, which is presumably why the body doesn't make an immune response to it and doesn't think it's foreign. We're probably lucky the body doesn't make an immune response to it because it might cause autoimmune disease. Well, they figured out that meninge B made proteins that other meninge have as well that are on the surface that they could use to make a vaccine. So reverse vaccinology, the idea that you start with the bug, predict in advance what's likely to be an effective vaccine. This has been used already, by the way, for Zika and many other things. This reverse vaccinology will be the future. So the big data, the precision medicine will be both on the host side and on the germ side. And last but not least, for infectious diseases, there's the vector. So for diseases like malaria and dengue, I don't have time to talk about it today, but it's very likely that genomics and precision medicine is going to lead to strategies to reduce or even eliminate some of the types of mosquitoes that cause malaria or dengue, and that strategy is likely to be the winning strategy to prevent Lyme disease, get rid of those ticks that spread Lyme disease, and you can get rid of the disease. So this is, at least in infectious diseases, precision medicine is having an impact on 3 levels: the host, the pathogen, and the vector. So I'll stop there, I think, and answer any questions. Yeah, done. Dr. Guerra.
No, this was an excellent talk. I think I learned a lot. I just had a comment that I agree are saying that the HSP, the molecular that is found in 2 patients, probably is just part of the story, because I think, like you say, there are these transcriptomes that probably play an important role. So you could have no mutation and still you don't really respond to be sure.
Sure, it could be regulated at some other level, but
Right,
absolutely
right.
true. Definitely the case that individual mutations only account for so much, even for monogenic disorders where we understand the mutation and what it does, cystic fibrosis, for example. There may be other genes that modify the phenotype. Tuberous sclerosis is a good example. We know what the mutations are in tuberous sclerosis, and yet if you look in the tumors from kids with tuberous sclerosis, there's a wide variety of other genes that are turned on or off. Like Jeff McKeegan in Grand Rapids is studying this for tuberous sclerosis.
So my other comment was your slide about Moore's law, about advancement in computer software has lagged behind advancement in our understanding of genomics.
Yes.
And we need this big computers to interpret that data. And I think, what do you think is going to happen? Because right now, I occasionally join precision medicine backgrounds that an Arbor has. And, right now, you look for, do the genomics on patients with who have refractory tumors or are in high risk for treatment failure. And it takes almost a month.
Yeah.
And so many, computer analysis to make sense out of that data. So where are we going?
So Dr. Guerra asked really the most important question of all, which is, how can we possibly assimilate all of these data? We're going to have the data, by the way, whether we want them or not. We're going to have gene sequencing on patients. Studies are being done now doing sequencing on every newborn in NICUs in certain places around the country. We're going to have a ton of data. The question is, how will we possibly be able to assimilate and understand that data? We need better computers and computer algorithms. We need more people trained in genetics. We need more genetic counselors. And right now, we talk out of both sides of our mouth. We're going to have this big precision medicine future, and yet we can't hire a geneticist because geneticists don't make money. Well, that's because they spend a lot of time sifting through a lot of complex data to help complicated patients. It's not the model that makes money. The model that makes money is I go set up urgent care and, incise some ingrown toenails. That makes more money than spending all day with a patient with a genetic disorder. So we've got to change that eventually. I agree with you. I think it's very worrisome that we'll have all these data and we may just flounder. We need algorithms. First of all, we need computer algorithms that help us figure out what part of the data is more important than others, because we're never going to have the wherewithal to analyze all the data as much as we might like. Take, for example, now those 2 mutations I mentioned, they did follow up those mutations. They tested them in a test tube. They showed that those cells don't respond to the HSV proteins, and they pretty much proved that probably was really the cause of the disease. However, in many cases, if I do whole genome sequencing on a child with autism, I may find a gene mutation that's been associated with autism 10 or even 15% of the time. I know that autism is up to 80 to 90% genetic based on twin studies, and yet you don't identify a specific gene mutation in most of those kids. However, if you find some polymorphism or some new mutation, even if it's a new mutation in a gene that's associated with autism, you don't know that that mutation is causing autism in that child. This is a big problem. The example I always give, which to me is the most chilling one, is cystic fibrosis. I mean, here's the gene. It was one of the first genes sequenced to find a mutation. Francis Collins. It's a gene we know more about than almost any other gene. We know a whole lot about what goes wrong with the protein with the different mutations now. We've got designer drugs that treat certain types of these mutations. And yet, not that many years ago, 15 years ago, when genetic testing for CF first went into prenatal testing first became available, the list of mutations that were thought to cause cystic fibrosis included one mutation which later was shown to be innocuous. So there were families that decided to terminate a pregnancy because they thought their child was going to have cystic fibrosis, and that child would have been normal. And that's a gene that we know a lot about. So you can imagine how difficult it is. You've got some new mutation in a new gene that's been associated with epilepsy in 3 kids. Now you've got another mutation in that gene in a child with epilepsy. Is that the cause of epilepsy in that child? I don't know. So we have to be a lot better. Actually, 2 of the people we just hired to start in Grand Rapids in precision medicine in February and April do try to figure this type of thing out. They take mutations, they set them in a test tube, they do computer modeling, they can put them in mice, they can try to figure out, but that's laborious, right? To do that for every new mutation to figure out, is this likely to be pathogenic or not? We need better predictive tools. And we need more sequencing. A lot of people say, well, once we have 50,000 more kids sequenced, then we'll have enough data to say if it means anything or not. It's just fine in that one child. There is a problem, though. The problem is all of you here who are physicians learned in medical school that the best type of experiment to do was a randomized controlled trial. We took 1,000 patients and gave them drug A and 1,000 patients and gave them drug B. And if 900 did better in A and 800 did better in B, you concluded drug A is better than drug B. And that's true for the population. But if you look at that study, there were patients in group B that responded very well to that drug, and some of them are not going to respond to drug A. And so the idea that just because it's better overall for the population means it's better for this child is not right. But there's a problem there. It's difficult to then do studies that involve an individual patient. You can take them on and off a drug, you can use a placebo, there are different things you can try, but it's not as powerful statistically as doing these large studies. And that's why some epidemiologists, like Dr. Hanna, caution us to say, be careful what you think you've learned from one patient with one mutation, but that might not be generalized to other patients. Nick.
Thank you, for very fascinating talk. I think one area that you mentioned, which Dr. Fanet, I think I've listened to Dr. Fanet talk about this, is the role of the environment.
Yes.
That, even with limitations, the environment, physical environment, social environment, emotional environment of the patient play roles, yes, whether or not the patient knows the disease or infection. What's your comment on that?
Yeah, so clearly environmental factors affect disease, right? Environmental factors do that largely because they affect gene expression, epigenetics. And they may cause direct toxicity, they may do other things, besides epigenetic phenotypes. But clearly environmental factors are important. And some of the skeptics about precision medicine, that's where they throw up their hands and they say, look at autism. I've given a version of this talk before, and I have the slides on here about precision medicine for autism, and the idea about making earlier diagnosis, earlier specific intervention. But as you say, even if you have a particular genetic mutation, the phenotype may vary quite a bit, and that's clearly affected by environmental factors. And if you look at people like Dr. Kassak at the Mind Institute at UC Davis has been studying environmental factors associated with brain development and autism for many years. I'm sure you know him, Jim. He's studied many environmental toxicants. And I've been to talks of his where he's outlined the data implicating dozens of agents in increasing the risk of autism in children. And so you start trying to figure out how in the world will we ever be able to compute the relative effect of exposure to this problem for this child with this genetic problem. I mean, you can immediately start thinking that the number of possible variations is greater, than the number of days that we'll ever have living on the earth as human beings, right? There's just so many varieties. It is challenging. However, people are trying to do it, and I would say the way they're trying to do it the best, and actually Ian Campbell, one of our new researchers in Grand Rapids, is working on this with somebody at Johns Hopkins, where they're looking at the effect of air pollution on neurodevelopmental outcomes and on autism. What they're doing in that is a lot of that is doing the same kind of geocoding that Mona and her colleagues, Rick Sadler, did in Flint. figuring out what parts of town were more likely to have the greatest lead exposure to water. Obviously, you can do the same thing. If you look at a metropolitan area, you can figure out where the levels of air pollution are lower or higher. People at Cincinnati Children's have done this, by the way, beautifully, looking at risk of asthma in the greater Cincinnati area. That when you come into the clinic, and they know where you live and what your zip code is and what your address is, they can already predict quite a bit about your risk of asthma before they know anything else about it, before they know any of your genetics. So I don't know how I think we learn from these studies what we need to do to reduce exposure to certain agents that are harmful to kids, but how we ever factor that in terms of what the relative contribution of this genetic mutation and that environmental exposure is, I don't know. I don't know if we'll ever get it. Roshni, last question. Everybody has to
That's
ask.
right.
Tina.
So besides the phenotype, genotype, is there any influence of race and gender?
Oh yeah, sure. There's also an influence of race and gender. There's someone in the veterinary school here who's doing fascinating work on inflammatory responses and the role of mast cells. And he's learning that the imprinting of mast cells between boys and girls early on in life is totally different. The gene expression patterns are different in mast cells during development, and therefore not surprisingly different during disease. And it's not just X chromosome related. It turns out there are a lot of indirect effects that probably have to do with hormonal regulation and genetics. Yes, there are clearly effects of race and gender and other things. So it's a very complicated situation. We've got a lot to learn, and we really do need a big data approach to ever figure it out. We're not going to be able to generalize from that one. All right, thanks everybody.